Hello, good morning everyone. A very good morning to all of you. Do you hear me? Am I visible to all of you? Please write in the box. Okay, let's start another topic of analytical biochemistry, that is lipoproteins and lipid profile in disease diagnosis. Again, it is very important clinically. So today's topic is lipoproteins and lipid profile in disease diagnosis. As you all know, lipid profile is very important in various diseases. First, we will take lipoproteins. That is a part of Unit 1, that is General Veterinary Biochemistry. So, what are the lipoproteins? What is the definition of lipoproteins? Lipoproteins are, please write in the box, lipoproteins are simple protein or conjugated or derived proteins. Please write in the box whether lipoproteins are conjugated, simple, or yes, these are conjugated proteins. Okay. Lipoproteins are molecular complexes that consist of lipids and proteins, as you all know. So these are conjugated proteins. They function as transport vehicle. It is a very important point that they function as transport vehicles for lipids in blood plasma. Lipoproteins deliver the lipid components, that is cholesterol, triacyl glycerol. What is another name of triacyl glycerol? Please write in the box. Triacyl glycerol. Traditional name of triacyl glycerol. Yes, neutral fat, and TG. TG is triglyceride or triacyl glycerol to various tissues for utilization. So here these are conjugated proteins and they function as transport vehicles for the lipid in blood plasma. Two important points in this slide. First is these are conjugated proteins and another is they function as transport vehicles for lipids in blood plasma. Lipids absorbed from the diet and synthesized by the liver and adipose tissues must be transported between various cells and organs for utilization and storage. Lipids are insoluble in water, you all know. The problem of transportation in aqueous plasma is solved by associating non-polar lipids. So lipids are polar or non-polar. Please write in the box. They are, yes. Am I audible to all of you? Please write in the box. Yes, yes. Okay. So lipids are insoluble in nature or insoluble in water. Now, the main problem of transportation in aqueous plasma is solved by associating non-polar lipids, triacylglycerol and cholesterol esters with amphipathic lipids. So what are those amphipathic lipids? Please write in the box. Yes. What are those amphipathic lipids? Please write in the box. We have already studied these lipids and their classification. These are phospholipids and cholesterol. So lipids are insoluble in water, you all know, and the problem of transportation in the aqueous plasma is solved by associating non-polar lipids like triacylglycerol and cholesterol esters with amphipathic lipids. Please take care of these two words, cholesterol esters and this cholesterol. So here it is triacylglycerol with cholesterol esters and with amphipathic lipids, phospholipids and cholesterol and the proteins to make water-miscible lipoproteins. What is the structure of lipoproteins? We will discuss in detail. A lipoprotein basically consists of a neutral or non-polar lipid core with triacylglycerol TG or cholesterol ester surrounded by a single surface layer or coat shell of amphipathic lipids, as I said earlier, that is phospholipids, apoproteins, and cholesterol. This is free cholesterol and this is cholesterol ester. The polar portion of amphipathic phospholipids and cholesterol are exposed on the surface of lipoproteins so that lipoprotein is soluble in aqueous solution, as I discussed in my previous slide. The protein moiety of a lipoprotein is known as apolipoprotein or apoprotein. Some apolipoproteins are integral and cannot be removed, like ApoA and ApoB, whereas other can be freely transferred to other lipoproteins, that is called as peripheral or ApoC, and examples are ApoC and ApoE. So mainly B and ApoA and ApoB are integral apoproteins and these are peripheral, can be freely transferred to other lipoproteins. There are two types of apolipoproteins or apoprotein. First is integral and another is peripheral. So structure of lipoproteins are, we can see here in this diagram, this is integral apoprotein, can't be separated. This monolayer of phospholipid and cholesterol, and this core cholesterol esters and TGs in this core, and these are peripheral apoprotein. So these are integral apoproteins, these are peripheral, monolayer of phospholipid and cholesterol, these are amphipathic in nature, and these are non-polar cholesterol esters or cholesterol esters and TGs, makes the core of this lipoprotein. So now is it clear to all of you? We will see in another slide this peripheral apoprotein, ApoC. This is monolayer of phospholipid and free cholesterol. You can see here. Likewise, this phospholipid and free cholesterol, and in the innermost you can see here cholesterol ester and this triacylglycerol or TG. So these cholesterol esters and triacylglycerol make core of this lipoprotein, and this is monolayer of phospholipids and free cholesterol. This is the integral apolipoprotein, as I mentioned earlier. This is integral ApoB and this is peripheral. As I said earlier, this is ApoB and this is peripheral ApoC. ApoC and this is ApoB. So now is it clear to all of you the whole structure of lipoproteins? Please add in the box. This is monolayer of phospholipids and cholesterol, free cholesterol, amphipathic in nature, and TG and cholesterol ester make the core of the lipoproteins. This is peripheral apoprotein or apolipoprotein. Example is ApoC, and this is integral. This is peripheral and this is integral apolipoproteins. What is the classification of lipoproteins? Lipoproteins can be classified in three ways. First is based on density. You all know kilomicrons, very low density lipoproteins, VLDL, low density lipoproteins, LDL, high density lipoproteins, HDL, separated by ultracentrifugation. Another classification or another category is based on electrophoretic mobility. Alpha, pre-beta, beta, broad beta lipoproteins. Mobility of lipoprotein is mainly dependent upon protein content. Please remember this sentence. Mobility of lipoprotein is mainly dependent upon protein content. Alpha migrates with alpha globulin, contains HDL. while beta migrates with beta globulin contains LDL. So alpha HDL and beta LDL. Another category is based on nature of apoprotein content like ApoA, ApoB, that is beta 100, beta 48, ApoE, etc. So lipoproteins can be classified in three ways: based on density, based on electrophoretic mobility, and based on nature of apoprotein content. You can see in this slide very clearly this density 0.95 skylomicron. First, density is lowest. You can see here lipoproteins with high lipid content. So chylomicrons contain high lipid with low density, that is 0.95, and larger size. You can see here this is largest in size, and those with high protein content like HDL. Here you can see high protein content sediment easily, have compact size is smaller, and have a high density. So is it clear to all of you? By this slide, you can easily classify these lipoproteins in various categories like HDL, LDL, IDL, VLDL, and chylomicrons. Okay, is it clear? Chylomicrons contains high lipid, HDL contains high protein. So lowest density, and this HDL have highest density, but size is smaller. Here size is largest. Okay. Classification of lipoproteins. 4 major classes of lipoproteins are identified in human plasma based on their separation by electrophoresis. So you can see here highest mobility, as I earlier said, higher protein content will move faster towards the anode. You can see here. If protein is higher, this moves faster towards anode. You can see here chylomicron origin, LDL pre-beta, LDL beta lipoproteins, VLDL pre-beta lipoproteins, and then HDL alpha lipoproteins. Higher protein content, HDL contains highest protein, so they move faster towards the anode. So is it clear to all of you? And one another category is IDL. You can see here this IDL. So major 4 and 5 categories, 5 major classes of lipoproteins are identified. Chylomicrons at the origin, then beta lipoproteins, LDL, VLDL pre-beta lipoproteins, and HDL alpha lipoproteins. So if protein content is higher, then they move faster towards the anode. So it is clear? Please write in the box, am I audible to all of you? And this classification of lipoprotein, have you understood? Okay. This is the table which you have to remember, contains characteristics of human plasma lipoproteins. These are the characteristics you can see here. As I discussed earlier, electrophoretic mobility, chylomicron's origin, VLDL pre-beta, LDL beta, HDL alpha. Then density, lowest density, then this range 0.96 to 1.006, LDL 1.006 to 1.063, and HDL 1.063 to 1.21, having highest density. Diameter or size, you can see here 100 to 1000, 30 to 90, 25, 20 to 25, and this 10 to 20 is lowest, compact in size. Apoproteins, you all know, A1, A2, B48, VLDL, B100, C1, C2, C3E. LDL contains B100. Here you can see B48, chylomicron B48, and this LDL B100. And this HDL A1, A2, C1, C2, C3, DE. It is important because chemical composition is entirely different. All the four categories of lipoproteins, like proteins, chylomicron contains 2%, it is in percent approximately, VLDL 10, LDL 20, as I said earlier, SDL contains highest protein, while lipid content is lowest. Chylomicron 98% means highest lipids, total lipids, VLDL 90, LDL 80, and SDL 60. Likewise, further, what is the composition of lipid? You can see here lipid components in percentage. TG triacylglycerol 88%. So chylomicrons contain 88% TG. Then VLDL 55, LDL 12, and lowest is with SDL. Cholesterol of free or ester, chylomicron 4%, 24%, 59, and 40%. Phospholipid is highest in SDL. So SDL contains highest phospholipid and chylomicrons lowest. Free fatty acid, you can see 111 in VLDL, LDL, and SDL. So now, is it clear to all of you? Proteins, highest proteins, SDL, highest lipid, chylomicron, TG highest, chylomicrons, but phospholipid lowest, SDL contains highest phospholipids. So is it clear to all of you? The characteristics of lipoproteins, please write in the box. So which lipoprotein contains highest phospholipids? Please write in the box. Which phospholipid contains highest HDL? Please write in the box which phospholipid, which HDL contains. Okay, no. Chylomicrons contains highest TG while lowest phospholipids, and HDL contains lowest TG and this highest phospholipids. Okay, so which lipoproteins contains highest phospholipid? Answer is HDL. Okay, now is it clear to all of you? HDL contains highest phospholipids while lowest TG. Okay. So we will discuss one by one. Chylomicrons, they are synthesized in the intestine and transport exogenous or dietary triacylglycerol to various tissue. This is the function of this chylomicrons. They are synthesized in intestine. Please remember, they are synthesized in intestine and transport exogenous or dietary TG to various tissues. Chylomicrons remain at the origin, as I discussed earlier, since it consists of highest 99% quantity of total lipids and lowest 1% concentration of protein. There you can see approximately this 99% and this protein. The chylomicrons are the least in density and the largest in size amongst the lipoproteins. Very low density lipoprotein, that is VLDL, they are produced in liver and intestine and are responsible for the transport of endogenously synthesized triacylglycerol. Please remember this word endogenously, because we have discussed that chylomicrons transport exogenous or dietary triacylglycerol to various tissues. Here it is VLDL transport endogenously synthesized TG. VLDLs with less protein content than LDL, you all know, more faster than LDL. This is due to nature of apoprotein present. Another category, it is intermediate density lipoproteins, that is IDL, are derived from catabolism of VLDL with a density ranging intermediate between VLDL and LDL. So this is between very low density and low density lipoprotein, ranging between 1.006 to 1.019, and the mean diameter or size is between 25 to 50 nanometer. Low density lipoprotein, LDL, they are formed from VLDL in the blood circulation representing a final stage in the catabolism of VLDL. It is an important feature representing a final stage in the catabolism of VLDL. They transport cholesterol from liver to other tissues. Please remember, they transport cholesterol from liver to other tissue. Here you can see chylomicron transport exogenous TG to various tissue. Then VLDL endogenously synthesized, transport endogenously synthesized TG, and then LDL transport cholesterol from liver to other tissues. And high density lipoprotein HDL, they are mostly synthesized in liver. Three different fractions, HDL 1, 2, 3, can be identified by ultracentrifugation. HDL particles transport cholesterol from peripheral tissue to liver. This is called as reverse cholesterol transport. This is called as reverse cholesterol transport. It means HDL particles transport cholesterol from peripheral tissue to liver. Please remember, LDL transport cholesterol from liver to other tissues, and here HDL transport cholesterol from peripheral tissue to liver. That is called as reverse cholesterol transport. So it is involved in reverse cholesterol transport. HDL involves in reverse cholesterol transport, also involved in VLDL and chylomicrons metabolism. So, is it clear to all of you functions of lipoproteins? Please write in the box. So, which lipoprotein is involved in reverse cholesterol transport? Please write in the box. Which lipoprotein is involved in reverse cholesterol transport? Means transport cholesterol from peripheral tissue to liver. Okay, good. Free fatty acids and albumin. Free fatty acids in the circulation are in bound form to albumin. You all know each molecule of albumin can hold about 20 to 30 molecules of free fatty acids. Free fatty acids and albumin complex, although not a lipoprotein, is an important lipid fraction in serum and is the fastest moving fraction and cannot be separated by electrophoresis. We have studied this free fatty acid albumin complex in genobiotics or metabolism of genobiotics also. So here these are the lipoproteins, these are apoproteins of these lipoproteins, and these are the functions of these lipoproteins. Chylomicrons, you all know APO4, ApoB48, ApoC, ApoE transport TGs from intestine to liver. TG, please remember this word TG, triacylglycerol. This VLD transport, VLDL ApoB100, here it is ApoB48 and it is ApoB100. Transport TGs from liver to adipose tissues or other tissues. Ideal intermediate between VLDL and LDL, as I discussed earlier. LDL, this ApoB48, transport cholesterol. This is an important point, cholesterol to peripheral tissues, and HDL involves in reverse cholesterol transport, absorb cholesterol from peripheral tissues and transport it to liver, reservoir for exchange of lipoprotein in VLDL and chylomicrons metabolism. Here it is ApoA, ApoC, ApoE and ApoD. So now is it clear to all of you? You can see here also in this table, you can see VLDL B100, LDL B100, chylomicron B48, and these are the A1, A2, the apoproteins of HDL. So again, you can see here ApoA, APO48, this VLDL ApoB100, and these are the functions of these lipoproteins. Please remember this table carefully. So Now we will discuss apoproteins or apolipoproteins. The protein components of lipoproteins are called apolipoproteins or simply apoproteins. The major apolipoproteins of HDL, that is alpha lipoprotein, are designated as capital A, and the main apolipoprotein of LDL, that is beta lipoprotein, is apolipoprotein B, that is B100, which is found only in VLDL. It is found only in VLDL. You can see here. Chylomicrons contains truncated or short form of ApoB, that is B48, that is synthesized in the intestine, while B100, that is found in VLDL, synthesized in liver. ApoE is found in VLDL, HDL chylomicron, and chylomicrons remnants. So these are apolipoproteins. As I said, HDL contains apolipoprotein A, LDL contains apolipoprotein B, and this is B100, which is found also in VLDL. Chylomicron contain truncated form of ApoB, that is B48, is synthesized in intestine, while beta 100 or B100 is synthesized in liver. What are the functions of apoproteins or apolipoproteins? They perform the following functions. First important function is act as structural components of lipoproteins, as you saw in previous slides, as the structural components of lipoproteins, like ApoA, ApoB, recognize the cell membrane surface receptors, act as ligands. Is the another function, recognize the cell membrane surface receptors, and another function is which activate enzymes involved in lipoprotein metabolism, acts as cofactors. So structural components, ligands, recognize the cell surface receptors and act as cofactors, activates enzyme involved in lipoprotein metabolism. Functions of apolipoproteins or apolipoproteins, they can form part of the structure of lipoprotein, as we have discussed. Examples are ApoB, structural component of VLDL and chylomicrons. So chylomicron contains which lipoproteins? Please add in the box. Chylomicron contains, or chylomicrons contain which apolipoprotein? Yes. B, ApoB 48. They are, yes, good. They are enzyme cofactors. C second for lipoprotein lipase, A1 for lecithin cholesterol acyltransferase, LCAT is an important point we will discuss in detail. Or enzyme inhibitors, ApoA2 and ApoC3 for lipoprotein lipase, and ApoC1 for cholesterol ester transfer protein. So these are the enzyme cofactors. They play as cofactors. They act as ligands for interaction with lipoprotein receptors in tissues like ApoB100 or ApoE for the LDL receptors, and APO1 for the HDL receptors. So it is very important that these functions of apoproteins or apolipoproteins are important in lipid metabolism. Like you can see as a structural component of lipoproteins, as ligands, as cofactors. In detail, you can see here enzyme cofactors like C second or A1 for LCAT and C second for lipoprotein lipase, LPL, and this act as enzyme inhibitors. Examples are ApoA second, ApoC third for lipoprotein lipase, and ApoC1 for cholesterol ester transfer protein. Act as ligands for interaction with lipoprotein receptors like ApoB100 and ApoE for LDL receptors, and ApoA1 for HDL receptors. These are the ligands, and these are cofactors, and these are the structural components of lipoproteins. So we will move on lipoprotein metabolism. First, transport of exogenous lipids. It is called as chylomicron processing. As I said earlier, transport of exogenous dietary lipids. So what is the function of chylomicrons? Please write in the box. What is the main function of chylomicron? Please write in the box what is the main function of chylomicron. Yes, to transport the exogenous lipid, mainly TG. So is it clear? Okay. It includes these 4 steps: absorption of dietary lipids, nascent chylomicrons, and lastly, formation of chylomicron remnants. So these include 4: transport of exogenous dietary lipids, mainly TG. This is also called as chylomicron processing, includes absorption of dietary lipids, nascent chylomicrons, and the formation of chylomicron remnants. Another is transport of endogenous lipids. First is exogenous and another is endogenous lipid via ApoB100 lipoprotein guided system and via APO1 governed lipoprotein mechanism or system. So you can see here the full metabolism of lipoproteins. First, the metabolism of chylomicrons. Chylomicrons or nascent chylomicrons are synthesized in the small intestine during the course of fat absorption. The first step of the metabolism, absorption of dietary lipids. Chylomicrons contains lipoprotein or apoprotein B48 and mostly TG. ApoB48 name is given since the absorption contains 48% of protein coded by ApoB gene. APO100 found in LDL and VLDL, but ApoB488 is found in chylomicrons. Chylomicrons are produced when nascent particle combined with ApoC second and ApoE derived from SDL. This is the important line of this whole slide, that chylomicron are produced when nascent particle combined with nascent chylomicron combined with ApoC second and ApoE derived from this HDL. The liver synthesize nascent VLDL containing ApoB100, which are rich in TG and cholesterol, and circulating SDL donates ApoC second and ApoE to convert nascent VLDL to VLDL. So you can see here SDL gives ApoC2 and ApoE to nascent chylomicron, and this circulating HDL also donates ApoC second and ApoE to convert nascent VLDL to VLDL. What is the role of lipoprotein lipase? You all know the enzyme lipoprotein lipase is present in the capillary wall of adipose tissue, cardiac and skeletal muscle besides other tissues. It hydrolyzes a portion of triacylglycerols or neutral fat or TGs present in chylomicron and to VLDL liberate free fatty acids and glycerol. Lipoprotein lipase is activated by ApoC second. It is an important point that it is activated by ApoC second. Uptake of chylomicron remnants by liver. So as the triacylglycerol of chylomicrons and VLDL are degraded, they lose the ApoC second, which is returned to S-dual. As I told you earlier, that this chylomicrons are produced when nascent particles combined with this ApoC2 and ApoE derived from SDL. So it is returned to SDL. Again, ApoC is returned to SDL, and the remaining particle is called as chylomicron remnant. The chylomicron remnants are taken up by receptors present on the hepatocytes of liver. So you can see here chylomicron by this SDL, ApoA and ApoC, TG in the presence of LPL, that is lipoprotein lipase, converts into free fatty acid and glycerol, and this chylomicron remnant, you can see this LDL receptors here, remnant receptors in the liver. Please write in the box what is chylomicron remnant. Please write in the box what is chylomicron remnant. When ApoC second, which is returned to SDL, Again, the remaining particle is called as chylomicron remnant. This figure shows the metabolism of chylomicrons. You can see here, this is HDL. HDL gives ApoC2. Here you can see ApoE, and this whole you can see here, this is the gut, this is liver, and this LPL, lipoprotein lipase, converts into, you can see here, fatty acids and glycerol, and this again return to ApoC2, and here it is returned to HDL again, and this is called as chylomicron remnant. And this is, you can see here, remnant receptors here. So this act as ligands. You can see here. As I told you earlier, that which is returned to ApoC2 returned to HDL, this is the particle is called as chylomicron remnant. So now is it clear to all of you? You can see here in this diagram, HDL gives this ApoC2, and then this is called as, combines with this ApoC2 and ApoE, and this is again converts into nascent chylomicron, converts into chylomicron, and this chylomicrons by the action of LPL, you can see here, converts into fatty acids and glycerol, and it is ApoC2 return to HDL, is returned to HDL again, and this is called as chylomicron remnants. And this is liver here. These are the remnant receptors. So please write in the box, have you understood the whole mechanism of this metabolism of chylomicrons? Please write in the box. As I told you in my previous slide, this contains 4 steps: absorption of dietary lipids, nascent chylomicrons, and again chylomicrons remnants. So next point is conversion of VLDL to LDL. During the course of VLDL metabolism, intermediate density lipoprotein IDL is formed, which loss ApoE and get converted to LDL. The Apo is returned to HDL. Please again take care of this point. This which lose ApoE and get converted to LDL, and the Apo is returned to HDL. LDL contains high cholesterol, as you know, free or esterified, and less triacid glycerol. So it contains high cholesterol. Here I mentioned circulating HDL donates ApoC2 and ApoE to convert nascent VLDL to VLDL. And VLDL to LDL. Cholesterol ester transfer protein, CETP. What is this? CETP is synthesized in liver, and it facilitates the exchange of components between different lipoproteins. Cholesterol ester transfer protein can transfer cholesterol esters from HDL to VLDL or LDL in exchange for TG. You have to remember this line. This cholesterol ester transfer protein, CETP, transfer cholesterol esters or cholesterol ester from HDL to VLDL or LDL in exchange for TG. LDL receptors and supply of cholesterol to tissues. The most important function of LDL is to supply cholesterol to the extrahepatic tissues. As I said earlier, the LDL particle binds to specific receptor pits identified as glycoproteins on the cell membrane. The shape of the pit is stabilized by a protein called clethrine. Please remember this word clethrine. The shape of the pit is stabilized by the protein called lethrine, and ApoB100 is responsible for the recognition of LDL receptor site. As I told earlier, this is the function of apolipoproteins or apoprotein B. So ApoB100 is responsible for the recognition of LDL receptor sites. What is the metabolism of SDL? This is high density lipoprotein are synthesized in liver as discoidal particles, nascent HDL. They contain free cholesterol and phospholipids, mostly lecithin. So, what is lecithin? Please write in the box. Please write in the box. What is lecithin? What is lecithin? Please write in the box. This is a phospholipid. And apoprotein A, C2, E, etc. They contain free cholesterol and phospholipid mainly. As I told you, just SDL contains highest phospholipid. So role of LCAT in SDL metabolism. It is LCAT. LCAT, what is LCAT? Lecithin cholesterol acyl transferase enzyme. The plasma enzyme lecithin cholesterol acyl transferase, that is LCAT, plays important role in SDL metabolism. The plasma enzyme lecithin cholesterol acyl transferase, that is LCAT. This enzyme is lecithin cholesterol acyl transferase, catalyzes the esterification of free cholesterol by fatty acids of lecithin to form cholesterol ester present in the extrahepatic tissue and transfers to SDL. So lecithin plus cholesterol here forms lysolecithin and cholesterol ester. In the presence of the enzyme LCAT, it esterifies this cholesterol, free cholesterol, and form cholesterol ester. APOprotein A promotes the activity of LCAT. This is an important point. SDL also accepts free cholesterol from other lipoproteins in circulation and cell membrane of peripheral tissues. Free cholesterol SDL undergoes LCAT catalyzed esterification, as I discussed in the previous slide. Due to the addition of cholesterol, SDL particle become spherical. The SDL particle with cholesterol ester trapped inside enter the hepatocyte by a receptor-mediated endocytosis. In the liver, the cholesterol esters are degraded to cholesterol, and the later is utilized for the synthesis of bile acids and lipoproteins or excreted into bile. As you know that in the liver, cholesterol esters are degraded to cholesterol, and this cholesterol is utilized for the synthesis of bile acids and lipoproteins or excreted into bile. So you can see the whole metabolism of SDL. This liver, you can see here bile acids and cholesterol in bile, APOprotein A, C2E, P is for phospholipid, E cholesterol. This is discoidal in nature, nascent SDL, and by the addition of this, you can see here this SDL is now spherical. A, C2 and E, you can see in the previous slide, due to addition of cholesterol, SDL become spherical. PC and CE. And by the enzyme this LCAT, you can see the extrahepatic cholesterol here, this addition, and this PC and CE cholesterol esters, and this now this is spherical. Due to the addition of cholesterol, SDL particle becomes spherical, and this SDL particle with cholesterol ester trapped inside enter the hepatocyte by the receptor-mediated endocytosis, and again this endocytosis goes to the liver. You can see here, and in the liver, the cholesterol esters are degraded to cholesterol, and this cholesterol is utilized for the synthesis of bile acids and lipoproteins or excreted into bile. So, Is it clear to all of you the whole mechanism or mechanism of metabolism of SDL or the whole metabolism of SDL, that is high density lipoprotein? Please write in the box. This extra hepatic tissue is cholesterol C. You can see here this discoidal nascent SDL, and then again this combines with cholesterol, extra hepatic tissues, and then this is now spherical in nature, and then this goes to by endocytosis goes to liver. Now is it clear to all of you the metabolism of SDL? Please write in the box. This is discoidal, this is spherical. Please write in the box. Am I audible to all of you? Am I visible? Okay. Nitesh Chaudhary, have you understood the metabolism of SDL? Nitesh, do you hear me? Okay. Abhilasha, Ramlal Tandi, Tarun, Rahul Pawar, this is the whole metabolism of lipoproteins, including LDL, chylomicron, SDL, and VLDL. You can see here, this is small intestine, this is nascent chylomicron, then this chylomicrons, and again you can see here this chylomicron remains and goes to liver. Nascent VLDL by this APOC2, you can see here SDL also, this SDL mechanism, SDL metabolism, E, then extrahepatic tissues, this enzyme lipoprotein lipase, you can see here, goes to liver, and this LDL here, B100, this, and this goes to liver. So in this slide, you can see the whole metabolism of lipoproteins, including chylomicron, VLDL, SDL, and LDL. And this goes to, you can see here, liver, and this you can see here SDL by C2 APOprotein, here C2, here C2, and this by this SDL you can see here E, and then again it goes to liver. And this you can see 3 fatty acids and this glycerol to liver. This is a summary of formation and fate of lipoproteins. Chylomicrons is a transporter of dietary lipids or exogenous lipid, mainly TG, whereas VLDL is a transporter of endogenous lipid, mainly TG, you all know. LDL transport cholesterol to peripheral cells from liver, while SDL transport cholesterol from peripheral cells back to the liver. You can see in this slide, chylomicrons is a transporter of dietary lipid, and you can see here, and this VLDL is a transporter of endogenous lipid, mainly TG, this exogenous and this endogenous, and LDL transport, you can see here cholesterol to cell or cholesterol to liver. This is the function of SDL. So now is it clear to extrahepatic cholesterol to liver, and this you can see LDL cholesterol to cells from liver. So now is it clear to all of you? Summary of formation and fate of lipoproteins. Have you understood? So please write in the box what is the main function of LDL. Please write in the box function of LDL. Please write in the box function of LDL? Am I audible? Okay. What is the function of LDL? Low density lipoprotein. LDL transports which compound? Yes. You can see here LDL transport cholesterol to peripheral cells or extrahepatic tissues from liver. What is the function of HDL, or what are the functions of HDL? You can see here first important function is squanging action. HDL squanges extra cholesterol or ester from peripheral tissue by reverse cholesterol transport. So what is the definition of reverse cholesterol transport? Please write in the box. It is called, why it is called as reverse cholesterol transport? Yes. It returns the cholesterol to liver. Reverse cholesterol transport. HDL transport cholesterol from peripheral cells to liver. HDL serve as reservoir of apoproteins. They accept apoprotein C2 and E and donate the same to other lipoproteins like chylomicrons and VLDL. So it is involved in the metabolism of chylomicrons and VLDL. The apoprotein C2 of HDL serve as activator of lipoprotein lipase, as I told earlier. HDL with the help of ApoE competes with LDL for binding site on the membrane and prevent internalization of LDL cholesterol in the smooth cells of arterial walls. HDL contributes its ApoC and E to nascent free LDL and chylomicron for receptor-mediated endocytosis. HDL stimulated prostacyclin synthesis by the endothelial cells, which prevent thrombus formation. Please remember the functions of HDL. And HDL also helps in the removal of macrophages from arterial walls. So HDL is very important for our body. HDL also helps in the removal of macrophages from arterial walls. These are the main functions: squanging action or reverse cholesterol transport, reservoir of apoproteins, then contributes in metabolism of chylomicrons and VLDL, and then STL stimulated prostacyclin synthesis by the endothelial cell, which prevent thrombus formation, and HDL also helps in the removal of macrophages from the arterial walls. This is the slide of reverse cholesterol transport, or we can see HDL mechanism, HDL metabolism. The cholesterol effects is brought about by esterification of cholesterol under the effect of LCAT, as I discussed earlier. The cholesterol ester rich HDL, HDL-L2. You can see here HDL2 is anti through squanger receptors, SRB1. Small intestine, phospholipid layer, you can see here, and this is liver, hepatic tissue, HDL2 and HDL3, this LCAT. This is the discoidal HDL, you can see, nascent, and again this spherical HDLs. So now we will come to disorders of plasma lipoproteins. Primary. There are two types of disorders, primary and secondary. First is primary, inherited disorder of lipoprotein in some individuals resulting in primary hypolipoproteinemias. They are due to genetic disorders or defects in lipoprotein metabolism and transport. So primary inherited disorders or genetic disorders. Secondary are the secondary acquired lipoprotein disorders are due to some other diseases like diabetes, mellitus, nephrotic syndrome, atherosclerosis, hypothyroidism, etc., resulting in abnormal lipoprotein pattern, which often resembles the primary inherited condition. So there are two types of disorders, primary and secondary. Primary hypolipoproteinemias and secondary acquired, which is related with other diseases like diabetes, mellitus, nephrotic syndromes, and atherosclerosis. So what is hyperlipoproteinemias? Elevation in one or more of the lipoprotein fraction constitute hyperlipoproteinemias. These disorder may be either primary or secondary. Number one, type 1. This is due to familial lipoprotein lipase deficiency. This enzyme is deficient in this disease, and the enzyme defect causes increase in plasma chylomicrons and Tg levels. Type 2A, this is known as hyper beta lipoproteinemia and is caused by defect in LDL receptor. So this is due to defect in this familial lipoprotein lipase deficiency, and this type 2A, which is also known as hyper beta lipoproteinemia, is caused by the defect in LDL receptors. Secondary hyperlipoproteinemia is observed and associated with, again, diabetes mellitus, hypothyroidism, nephrotic syndromes, etc. And this disorder is characterized by hypercholesterolemia. Deficiency of LDL receptors are deficient or a defect in LDL receptors results in the elevation of plasma LDL, hence plasma cholesterol. So this hypercholesterolemia. However, plasma Tg concentration remain normal. Deficiency of LDL receptors is observed in type 2 hyper-beta lipoproteinemia. This disorder is associated with very high risk of atherosclerosis, particularly of coronary artery. Next, third type of hyperlipoproteinemia is type 2B. Both LDL and VLDL increase along with elevation in plasma cholesterol and Tg, and this is believed to be due to overproduction of ApoB lipoproteins, ApoB, apoprotein or apolipoproteins. Fourth type third, this is commonly known as broad beta disease and characterized by the appearance of broad beta band corresponding to intermediate density lipoprotein IDL on electrophoresis. Type 4, this is due to overproduction of endogenous triacylglycerol with the concomitant rise in VLDL. This disorder is usually associated with obesity, alcoholism, diabetes mellitus, etc. Type 5, both chylomicron and VLDL are elevated. This is mostly a secondary condition due to this disorders such as obesity, diabetes, excessive alcohol consumption, etc. So these are hyperlipoproteinemias, primary and secondary. Type 1, type 2A, type 2B, type 3, type 4, type 5. This is hypolipoproteinemia, although low levels of plasma lipids within the normal range may be beneficial to the body. Very low lipid levels are undesirable. These are commonly associated with certain abnormalities. Very important, familial hypo-beta lipoproteinemia. It is an inherited disorder, probably due to an impairment in the synthesis of apoprotein B. The plasma LDL concentration in the affected individual is between 10 to 50% of normal value. This disorder is harmless and in the individuals have healthy and long life. This is familial hypo-beta lipoproteinemia. These are the conditions of hypolipoproteinemias due to the impairment of the synthesis of apolipoprotein B. Second is A-beta lipoproteinemia. This is a rare disorder due to defect in the synthesis of apoprotein B. It is characterized by total absence of beta lipoprotein in plasma. So it is called as A-beta lipoproteinemia. Defect in the synthesis of apoprotein B. Triacylglycerols are not found in plasma, but they accumulate in liver and intestine. Serum cholesterol level is low. A-beta lipoproteinemia is associated with decreased absorption of fat and fat-soluble vitamins. Impairment in physical growth and mental retardation are commonly observed in this A-beta proteinemia, A-beta lipoproteinemia. Third is familial alpha lipoprotein deficiency, that is called as Tangier disease. The plasma HDL particle are almost absent. This is called as familial alpha lipoprotein deficiency. So HDL contains which lipoprotein? Please write in the box. HDL contains which lipoprotein, which apoprotein, apolipoprotein or apoprotein? Please write in the box. This is alpha. Due to this, the reverse transport of cholesterol is reversely affected. This alpha, no beta, this is alpha lipoprotein deficiency. The plasma HDL particle are almost absent. Due to this, the reverse transport of cholesterol is severely affected, leading to the accumulation of cholesterol esters in the tissues. Can't be back to liver. An absence of apoprotein C2, which activates lipoprotein lipase, is also found. The plasma triacylglycerol TG levels are elevated in this condition. The affected individuals are at an increased risk of atherosclerosis. So this is the disorder of lipoproteins, or we can say show hyperlipidemia or hyperlipoproteinemia. Type 1, you can see here familial hypochylomicronemia, this type 2, this type 2B, type 3, type 4, and type 5. And these are the defects and these are the abnormalities. You can see here again this hyperlipoproteinemia type 1, type 2A, type 2B, 3, 4, 5. Increased plasma lipoprotein chylomicron, LDL, LDL, and VLDL. IDL in 3rd, VLDL in 4th, chylomicron and VLDL are in 5th. This level of increased plasma lipids, mostly TG in first type, cholesterol in second A, TG and cholesterol in second B, TG and cholesterol in 3rd, TG in 4, TG in 5. These are the metabolic defects. These are the risk of atherosclerosis. May increase very high in second A, very high, mostly in coronary artery. Very high, you can see here, mostly in peripheral tissue. And this suggested treatment. Low fat diet, low cholesterol fat diet, low fat, low caloric diet, low fat and low caloric diet. What is fatty liver? In certain conditions, lipids, essentially the triacylglycerols or TGs, accumulate excessively in liver, resulting in fatty liver. So what is fatty liver? Please write in the box. In certain conditions, lipids or fats, especially the TGs, accumulate excessively in liver. It is called as fatty liver. Hepatic triacylglycerol synthesis provides the immediate stimulus for the formation and secretion of VLDL. Impaired VLDL formation or secretion leads to non-mobilization of lipid components from the liver, resulting in fatty liver. In the normal liver, Kupffer cell contain lipid in the form of droplets. You all know in fatty liver, droplet of TGs are found in the entire cytoplasm of hepatic cells. This causes impairment in metabolic functions of liver. Fatty liver is associated with fibrotic changes and cirrhosis, liver cirrhosis. Fatty liver may occur due to two main causes: increased synthesis of TGs and impairment in lipoprotein synthesis. What are lipoprotein agents? Lipoprotein agents. These are the substances, the deficiency of which causes fat to accumulate in liver, mainly TG. Agents such as choline, inositol, methionine, betaine, glycine and serine, and other essential amino acids, essential fatty acids, antioxidant vitamins, vitamin B12, folic acid, and synthetic antioxidant, which have the apparent effect of removal of fat from the liver cells and thus prevent the formation of fatty liver, are called as lipoprotein agents. So, what is the definition of lipoprotein agent? Please write in the box. What is the definition of lipoprotein agents? What is lipotropic agent? The agent which prevent the formation of fatty liver. Am I audible to all of you? Please add in the box examples of lipotropic agents. What are the examples of lipotropic agents? They prevent the formation of fatty liver. Then another lipid profile, it is hypercholesterolemia. Increase in plasma cholesterol concentration is known as hypercholesterolemia and is observed in mainly these disorders like diabetes mellitus, due to increased cholesterol synthesis since the availability of acetyl coenzyme A is increased in hypothyroidism, myxedema, in obstructive jaundice, various nephrotic syndrome, increase in plasma globulin, concentration is the characteristic feature of nephrotic syndromes, and then cholesterol elevation is due to increase in plasma lipoprotein fractions in this disorder. Hypercholesteremia is associated with atherosclerosis and coronary heart disease that is called as CHD. So you all know the impact of cholesterol. More specifically, LDL cholesterol is positively correlated, where SDL cholesterol is negatively correlated with CHD. LDL cholesterol is positively correlated with CHD, means when LDL cholesterol increases, the possibility of CHD also increases, where when the concentration of SDL cholesterol increases, the possibility of CHD lowers or decreases. What is bad cholesterol and good cholesterol? Cholesterol is a natural metabolite performing a wide range of functions: membrane structure, precursor for steroid, hormones, bile acids. You all know the cholesterol in high concentration present in LDL is considered bad due to its involvement in atherosclerosis and related complications. Thus, LDL may be regarded as lethally dangerous lipoprotein. Small dense LDL, SDL, SD, small dense LDL is considered to be most dangerous fraction of LDL associated with coronary heart disease. On the other hand, SDL cholesterol is good since its higher concentration counteracts atherogenesis. SDL may be considered as highly desirable lipoprotein. So, what is bad cholesterol? Please write in the box. Cholesterol, which is present in LDL, is called as bad cholesterol, and which is present in or associated with HDL is called as good cholesterol. Hypocholesteremia, a decrease in the plasma cholesterol, although less common. Hyperthyroidism, pernicious anemia, malabsorption syndrome, hemolytic jaundice are of the some disorders associated with hypocholesteremia or the decrease in the plasma cholesterol. It is less commonly observed. So, have you understood the whole lipoproteins and lipid profile? Please add in the box. Am I audible to all of you? I will summarize the whole presentation in few minutes. Important points. So, these are the lipoproteins, characteristics of plasma lipoprotein. You can see here chylomicron at the origin, pre-beta VLDL, LDL beta, SDL alpha, density, diameter apoprotein. You can see here B48, VLDL B100, LDL B100, and ApoA. Protein highest in SDL, lipid highest in chylomicron, TG in chylomicron, lowest phospholipid in chylomicron, highest phospholipid in SDL. So chylomicrons transport TG from intestine to liver, VLDL transport endogenous TG to liver to adipose tissue. This is dietary or exogenous. Ideal intermediary between VLDL and LDL. LDL transport cholesterol to peripheral tissue, and SDL involves in reverse cholesterol transport, or you can say absorb cholesterol from peripheral tissue and transport it to liver. These are apolipoproteins. Functions of apolipoproteins are structural component of lipoprotein, act as ligands and cofactors. What is the mechanism of metabolism of lipoproteins? Transport of exogenous dietary lipids by chylomicron processing involves absorption of dietary lipids, nascent chylomicrons chylomicron remnants, and the transport of endogenous lipids by ApoB100 lipoprotein and by ApoA1 governed lipoprotein systems. This chylomicron, you can see here this chylomicron remnant. This C2, ApoC2, and again returns to HDL, and this is called as chylomicron remnant. Metabolism of HDL, role of LCAT, lecithin cholesterol acyltransferase converts into lysolecithin and cholesterol esters. Apoprotein A promotes the activity of LCAT. You can see here this combined with cholesterol esters hepatic tissues, and this spiral in shape, and now this is spherical, and again goes to liver by endocytosis, and this is called as reverse cholesterol transport. Here you can see chylomicron, VLDL, HDL, LDL, again this chylomicron remnants in liver. You can see here, and this led by lipoprotein lipase, free fatty acids, glycerol to liver. Summary of the formation of fate of lipoproteins. You can see here chylomicrons transports dietary lipids, VLDL endogenous lipid, mainly TG, LDL cholesterol to peripheral cells, and HDL transport cholesterol from peripheral cells to liver back. Functions of HDL, sponging action, reservoir for APOproteins, and then removal of macrophages. This is reversed cholesterol transport by LCAT. Disorder of plasma lipoprotein, primary and secondary. You can see here hyperlipoproteinemias 1, 2A, 2B, 3, 4, and 5. And this is fatty liver. What is fatty liver? Fatty liver is the condition when lipids, especially TGs, accumulate excessively in liver due to increased synthesis of TGs and impairment in lipoprotein synthesis. Lipotropic agents, which prevents the formation of fatty livers like choline, inositol, methionine, betaine, glycine, serine, and other essential amino acids. What is hypercholesteremia? Increase in plasma cholesterol in the condition of diabetes mellitus, hypothyroidism, obstructive jaundice, nephrotic syndromes. It is related with CHT also. Bad cholesterol. The cholesterol is in high concentration present in LDL is considered as bad, and with the HDL it is considered as good. And lastly, hypocholesteremia, when concentration of cholesterol decreases. This is not commonly observed in the condition of hyperthyroidism, pernicious anemia, malabsorption, hemolytic jaundice, and various other disorders. So, thank you very much. Thanks once again.